So
Okay.
today we have a talk by Tianyu, who is a participant and medalist of the Kaggle competition on the analysis of the single cell data. And thank you very much that you agreed to make it talk. Please start.
Okay. Thanks, Alexander, for the wonderful introduction. So hello everyone, I'm Tianyu Liu, and I'm a member of Team 1 only in this single cell data analysis competition. So our team member include Jianwu Zhou and Average Grants. So in today's presentation, I intend to talk about the overview of these problems, including some problems I think existing in this topic and the challenges we will face, and our design for the models we select to predict, and some additional functions we have made to explore the biological aspects, and the brief discussion, the summary of the presentation. And for the problem overview, we know that for our topic, we intend to predict the one modality data based on another type of modality. For example, to predict the RNA expression based on the DNA accessibility, and to predict the protein expression based on the RNA sequence data. So for this kind of topics, like we have 10 days data in total. So for some of this data, they are training data sets. For other of them, they are public tests and private tests. So our task is like to utilize the a cross-validation score based on the training data set and to predict the public test results, and then to use this public data set to fit our prediction for the private test. And the difference between the public test and private test is about the donors and the time series. Because we sequence the cell data from different time, and now we utilize different kinds of we use same techniques to sequence the cells from different days, and this is a kind of time series. So this is the first challenge I think in this kind of task. And the second one is about the selection of samples or donors, because we can see that in our public test data we only have one donor, I think, but in our private test we have 4 donors. So it means that our model should have the functions to, for example, catch the similarities among different donors. So this is the second challenge. And the third challenge is made by Rosen, I think. Rosen, okay, is another participator in this competition. And we can see that the cell proportions are different among the with this increment. Because this is a kind of cell differentiation process. We can see that with the time goes, the proportion of the green kind of cells, the proportion is become lower and lower, but for other cell types, they are becoming larger because this kind of cell types has been transferred into other type of cells. So this is the third challenge. So it means that if in the identical condition, like our model should simulate the 3 kinds of conditions. So in our method, we have considered about all these 3 problems. So our overall method can be concluded into the 3 components. The first thing is about the feature selection step. So it's about pre-processing. And we also utilize the neural networks-based models with the tree-based boosting models with the ensembling. And for the cross-validation method, we use 2 kinds of cross-validation strategies. The first thing is about the group threefold, which means that we split the group into different donors. For example, we use 2 donors to predict the results from 1 donor. And we also add a simple 5 voice to increase the variation of our results. And for the single cell RNA sequence part, I mean to use single cell RNA sequence data to predict protein data. For our data, we first perform the pre-processing step of feature selection for the single cell RNA sequence data part, which is known as the input part. And we can select some important genes. So because if we observe the name of the genes, we can find that there are some proteins which are included in such genes. We call such genes as protein coding genes. And we keep such genes in its original value. And we also run the SVD analysis for the whole single cell RNA sequence data set to select the important components. And then we can include the input data set into the neural network-based method and some gradient-based boosting decision tree-based method. So here's a law of thumb to select the gradient boost tree. So although we use 3 kinds of models here, like SG boosting, cat boosting, and LightGBM, so actually they have their different advantages. So they have their different application scenario. For example, if you only have the GPU, so it is recommended to use the LightGBM. And if you have GPU, you can use XG boosting with sorry, this is a typo. XG boosting method with the GPU acceleration. And if there are plenty of categorical features, for example, 0, 1 features in your training data set, you can choose to use catboost. And we have the predicted protein expression here based on our combined model, and we can assign different Ys for this prediction expression output based on the model ensemble. And for the targets here, we do not modify the targets. We only perform the standardization operation because this is mentioned by the computation holders. And for another part, the multi-omic part, we firstly perform the binarized operation based on single-cell ATAC sequence dataset because it can be treated a kind of denoising approach, and we will illustrate this point later. And here, like we intend to use multi-omic dataset to predict the single-cell sequence dataset. The dimensions of multi-omic dataset is very, very large. And for the single-cell sequence dataset, we also have plenty of genes. So it is very impossible for us to predict each gene value directly. So here we also perform the dimension reduction towards the targets part. We also use the SVD operation for the target dataset and save the loading matrix here. So now our target is to use some dimension reduction ATAC sequence data to predict the dimension reduction targets. So we also utilize the neural network and plenty of boosting decision tree-based method and then get the predicted results. So now we can modify the prediction results with our loading matrix and then perform the model ensembling based on weighted sum method to generate our final output. So another operation we have another approach we have applied for the single-cell ATAC sequence prediction is about the combination variation of the input and output data. So for example, we can split the train input. Because we can use SVD method to perform the dimension reduction for the train inputs, and we can choose to adjust the dimension of the SVD operation, like from 16 to 18, and then vary it by the combination of the input and target data sets, because we also perform the SVD for the output data. And based on this design, we have more than value variation types of our same model. So this means that we can generate better representation models, which means that these results will be a better estimation for the for the private data set. Yeah. And for the neural network methods, we don't utilize the simple MLP, so we actually add plenty of tricks here. I can summarize them into 3 key components. The first thing is residual net, so which means that I will connect the output of, for example, layer L1. I will connect the output of layer L2 with the layer L. So layer L2 is the previous layer of layer L1, and layer L is the future layer of layer L1. So which means that this is a kind of skip connection. So with this kind of mechanism, we can eliminate gradient vanishment problems existing in the neural network training. And the second thing is about activation function. So activation function is not chosen by abruptly or by your own idea. So we need to choose the activation function which can fit our target. For example, in here, our prediction output is a kind of standardized data. So we can find that there is a kind of neural network activation function which can also generate the output follows the standardized distribution. Here we choose to use SILU as an activation function. So in the paper of SILU, it proves that if we select the suitable parameter initialization method and suitable output conditions, the output of the SILU function will converge into a distribution with mean of 0 and variance of 1. So we prove some theoretical guarantee for this kind of application. And the third thing we have used is about the I believe operation. So it is a kind of state-of-the-art optimizer existing in the neural network optimization. We can see that in the 4 examples given by these authors, the I believe can always find the optimized points compared with the state of another type of well-known optimizers, ADAM and SGD with momentum regularization. And for some additional functions we have used, including how can we apply the biological concept into our application. So the first thing we have tried is about data binarization. Because we can see that for the single-cell ATAC sequence dataset, it is very sparse. So for each cell, we can divide it by the maximum value and perform the scaling function to transfer the matrix into a 0-1 matrix. So for the advantages, it's very obvious. We can see that for the UMAPS data before the data binarization, and the cell type, we cannot observe a clear borderlines existing in different cell types. But if we perform such such operation, we can observe such borderline of the difference existing in different cell types, which means that it can help us to solve the problems caused by the cell differentiation. Another type of additional functions is about data imputation. So the idea of data imputation is come from the matrix computation. It means that, for example, because we know that for single cell data set, it is very sparse. We have plenty of zeros here. So some of the zeros, they are meant as the true gene expression value of 0, but for others, they are caused by the technical issues. So we call them as fixed zeros. So to solve the problems caused by the fixed zeros, we need to rely on the data imputation. For example, matching is a kind of method to perform data imputation, which means that it will fill the matrix with 0 values as is based on its neighbor cells information. So after the magic performance, we can see that the original very sparse matrix become dense than before. But magic suffers the problems caused, we call it oversmoothing, because it may lead to plenty of false positive results here. So we also consider about another type of method known as deep impute. So deep impute is a denoising autoencoder based method for data imputation. But unfortunately, we have tried this method, but the result is not very optimistic. And the second thing we have tried is about the grafting network. Based on the inspired by the ideas from the magic, we can choose to construct the cell similarity matrix here, like to construct them based on the graph. For each node, its feature is about the gene expression, for example. And for the output graph, it will also be the same node, represent the same cell, but the expression will become the protein expression. But the result is not very optimistic, because I think there are two major problems existing in the graph neural network-based prediction process. The first thing is maybe we need to face a very, very large graph, right? Because we have plenty of cells here and we cannot drop these cells. The second thing is about oversmoothing. We know that because the graph neural network is based on the graph convolutional approach, it will try to catch the neighborhood information to perform the propagation. So as time goes, like it finally will converge, like all the nodes will share their one features if they have the age. So this is the oversmoothing problems. But I think this approach is worth trying in the future, because we have some another kind of powerful graph neural network. So here we only try the graph convolution network, I think. And for the last additional functions is about submodel optimization. If we plot the correlation graph for all the proteins we intend to predict, we can see that some of the proteins, they are easy to predict, right? But others, they are very difficult to predict. So I think this may be caused by the importance of such proteins. For example, CD41 and CD71, they are very important in the human blood cells, but for other proteins like CD83 or CD23, their functions are less important. So this inspires us to design different models to perform the prediction for different proteins. For example, in the proteins which are good based on our old models, we can keep them. But for other proteins, we can use a new model to predict such proteins. But we need to select a threshold, for example, for select point file, to distinguish the proteins which are easy to predict and difficult to predict. But I think this may be because the new model is not very powerful, so I still did not observe the score improvement based on such design. And the last thing we have tried is about the meta-embeddings. So we know that we have other meta information here, like gender. So it is implicit information, and the cell type, and this, they are explicit information. They are categorical data, but we can always transfer this data into a vector space based on the embedding method, for example, transformer or just the scapegram. So now we just use a very simple scapegram embedding method here to transfer the metadata information into the vector space data and then combine them with the input data. So we finally include this part into our training process or into our final output or final submission file to the computation because it can increase the variation of our model output. Right? And for the discussion part, like I intend to talk about some interesting findings we have found in this computation. Because like in our previous models, we actually have a very powerful model, like based on the public score, but they are not the best thing in our public score. So actually our best public score can always yield to the overfitting for the private score. So it means that the difference existing, I mean, the difference for the different donors and dates, they may be very, very large. We need to consider about how to estimate the difference between the private data set and public data set. So it seems that so far, there are not so many, I mean, for the top participators, even in this competition, I think they don't pay attention to this aspect. I think this is very worth it to explore. And there are also some other potential problems may cost, maybe can be used to solve the different, I mean, to minimize the difference between the private dataset output and public dataset output. For example, maybe we can choose to use another type of single cell pre-processing steps. We can choose them as we can drop the normalize and log 1p transform here. And for the feature selection, we can also utilize some manual void-based method to perform the feature selection, but it will be very time consuming. And we can also try some different loss function because, for example, some participators I know they have tried the huge loss or they use cosine similarity or cosine difference here, and it is also worth to try. And for the discussion part, it's another very interesting research is related to its downstream analysis. So for example, we can try to plot the protein expression here based on the marker proteins for each cell type. And if for example, for the HCA-CB1, it is a protein for the CD4T cell. And if we observe the high protein expression for CD3 cells in the T cell, CD3 protein in the T cell, which means that we have performed very good generalization for the CD3 protein here in the T cell. So we can use the plot like as a kind of variation approach to prove the functions of our model. And we can also use some gene to peak linkage discovery method, for example, SIGNAC, to find the linkage between the genes and the peaks to verify that if our prediction can really work. And so for other things that I have tried here is about the maybe exploration approach for the neural network method. So it is well known that there is a toolbox known as SHAP. It uses SHAP score to evaluate the importance of each feature. So its workflow is very easy to understand because we have the model like accept different features and then generate the output here. We have plenty of number of samples and the number of features here, and we can replace the tree explainer here like based on neural network explainers. We actually train a neural network explainer here to generate the SHAP values or the local explanations. So SHAP values come from the idea related to the game theory. So it evaluates the importance of the features of steps we have chosen in the game series. So now it's about the features. For example, if we run the SHAP analysis for my own method for the protein or for the for the statistic prediction part, and we can observe that there is a kind of features like 8, 16, 3, like they are positive correlated with our prediction results, and there are also other features maybe they are negative correlated with our prediction results. And I also observe that all of the features here, I mean the important features, they are the protein encoding features, right? But if we remove the other SVD results, I mean if we remove the SVD results for the whole data set, the prediction results will not be very identical. So this is a very interesting thing from my end, because I think this implies that the neural network method is also very, very complicated. Yeah, so maybe we can explore more things here. And just for a quick summary, so that in these problems involving the time series cross-validation scheme, and we need to choose the CV method very, very carefully because this is the only standard we can rely on to adjust our parameter. And in single-cell data related work, the models based on neural network can perform very well. So we have talked about these things in the previous slides, and I think of the top 3 participators, they have applied the neural network in their final submission. And for the model ensembling, it's a very effective solution to mitigate overfitting. So some bio-driven methods, they are very diverse and do not perfectly fit the application tag scenario. I think this reasons depends. Some methods, they don't have very, very good like assumption, like their bio-driven points may be wrong. But for some other methods, maybe we don't find the good parameters existing in such model. Yes, and the last thing that I think we need to enjoy is this kind of research process to learn some knowledge. I think it's will be pretty cool. Yes, thanks. I think that's all I want to say. Thanks.
Thank you very much. So it's a great talk. Okay, so everybody to welcome to ask questions.
Yeah.
And so now you can unmute yourself and ask question by voice. Now bonus anymore. or you can write a question in the chat. It's up to you. Okay, so may I ask you just to start the questions? So about the feature importance. So
okay.
So the first question, you use SHAP. You use
Yes.
it for all neural networks and for gradient boostings, or only for neural networks? So
yes. So thanks for the question. I only apply it to the neural network-based method, because I think for the tree-based method, like actually boosting or the boosting-based method, they have their own feature selection importance function.
Yes.
So we don't need to apply the. And actually, in the paper of the SHARP method, they have proved the consistency existing in their SHARP values and the feature importance in the tree-based method, I think.
Okay, so there is a question in the chat. Which library did you use for SHAP?
There are other. Sorry. I can maybe type the link to you. I don't know if there are other SHARP library here. I think it's only one official library in the GitHub. I think it's from Swedenborg's. I can type it. It's the first author of this method, I think.
Okay.
Oh,
thank you very much for sharing. So don't you have some plots of the feature importances? So how many features you think are important? Yes. And another question is about, so this feature importance is for all targets simultaneously, or it's just you can you have feature importances for each target separately, for each protein, you know, CD1, CD3.
Yeah, so that's a good question. Also, like for the first question, I think it's hard to say that so actually each feature, they have their own SHARP value, or they have their own importance, right? So we actually rank them based on this plot, and we can see that some of the I should say that all of the features like labeled by the authors of this method in this plot, like they can be treated as important features, I think, based on your scenario. And for the second question, I think because I personally don't predict the proteins one by one. I mean, I don't use like 140 models to predict. So I think this SHARP values represent the feature importance towards all of the proteins.
Okay, I see.
Yeah, because one reason I don't use the like one by one models is because I think because there exists something like the co-expression network in the protein regulation network. So each protein, their output may affect the output of another kind of genes. So this is kind of the co-expression network or expression network. So that's why I think we need to consider about the functions of the similarity functions or difference functions of all the genes that to perform the prediction. Yeah.
Okay, thank you. So now there are two more questions in the chat. The first question seems to be to me. You mentioned some magic earlier. Is it the same magic that Tianyu mentioned? That Elena asked. Yes, Elena, it's the same magic. Yes, it's the quite famous package for denoising the single cell data. Tianyu, maybe you can show this slide with the magic again.
Yeah, I think we are. This is the same thing as you said in the magic.
Yes, so we have also tried to use it with some partial success, I would say. So we tried to use magic, and we selected some features from magic features, not all of them, and some of them gave us a small improvement. Okay, so
okay, so for Alex's question, I think we actually have tried to merge activation function here, but we don't observe a very significant score improvement. And because we know merge function is a kind of exponential component, and it's a like combination between the soft plus, I think, with another function is exponent. So if we like use the exponent operation in our operation process, it will increase the calculation consuming. I'm not sure if yeah, so the running time will become longer. So that's why we don't use the merge function here. But you can try it. I think merge function also, I think you need to adjust the parameters, yeah, to achieve its best performance.
There is another question in the chat. Have you checked niche activation function?
Yeah. I'll just talk about it.
Okay, sorry.
Can I ask what cross-validation strategy did you use? Did you include day in it or?
Yeah, so I think the cross-validation we have tried here, like, is based on donors. Like, this is the initial idea, I think, in the early stage of this computation. Like, for example, if we have 3 donors in total, like our first cross-validation group is to use donor 1 and 2 to use to treat them as the training data set, and use the donor 3 as the test data set, and then we repeat this for the whole donors.
I see.
Yeah, so this,
thank you.
Yeah, this is the first cross-validation method we have tried. And the second thing is about simple file voice. It's just like I have 4 donors data, and then I just sample them, maybe like sample them into 4 training data set and 1 as test data set, and this simple k-fold. Yeah.
Okay, I see. Thank you. And about the important genes, how do you choose them?
Do you mean the importance here?
No. This slide in methods.
No.
You selected
The important genes. Yeah.
Yeah. How did you select them?
I think if we take a look at the important, if we take a look at the gene names of the single-cell sequence data set, we can see that there are some genes, they have their protein annotation, like behind these gene names. So it represents that these are protein encoding genes. So like their protein encoding, their encoding proteins existing in our prediction targets. So that's why we must include them, I think.
I see. Thank you.
You're welcome.
Colleagues, any other questions, please? So you are welcome to ask in chat or by voice, up to you.
Okay, hello. Thank you very much for the talk. And I'm wondering, I'm not very familiar to graph neural nets, but can you explain, please, if you have, for example, a text data, and you can build some graph, and on this graph you can apply GNN, but how you will from this structure, how you will understand the expression. For example, for the first task, when you on multi-ohm, when you have a text check and you're building graph, and how GNNs predict the expression from graphs. Can you please explain?
That's a very good question. So yeah. So like there are different kind of implementation for the GNNs. I think there is another talker, another presenter in the NearIPS competition, like they use the GNNs, but their rank is lower than us. I can send the links about their graph library to you later, because their graph is more robust than my current design. Like this is better than my my graph neural network design, and they will keep on updating their package. I can send this link to you later. But for my graph, the general idea is very similar to like magic, because we can use the for example, if we need to predict the protein expression data based on gene expression data, like here, and for each cell we have its gene expression profile, and we can use the similarity between this, for example, the profile of cell I and the profile of cell G to calculate the correlation, right? So based on correlation, like we have the Y's for each age, or we can use this correlation as a threshold to cut off the age to finally construct a graph. So like based on the profile expression value, we have adjacent matrix here. And after we have adjacent matrix, we also have the cell's gene expression. So we call them as feature for each node. For each cell, they are treated as node, and for the age, they represent the relation between different cells. And we can input this relation with the feature information into the grafting network to perform the to use graph Laplacian and graph convolution here, and to encode the features into a lower embedding representation, a lower dimension representation. And then we can use another type of layer. It works like a decoder design, and then to decode such information into the protein part. Yeah, so this is like my gene design. I can send this link to you. So give me a moment. I need to spend some time to find it.
Sorry, but the first picture cells. So the graph is based on something like a KNN graph or some similarity graph between the cells.
I mean, GNN is not GNN is not an approach to construct graph. GNN accepts the graph as the input, and it will generate the representation for the graph. So for my approach to construct the graph, I use the cell similarity matrix. Yeah.
So you use the KNN graph, so K nearest neighbors.
Yeah. KN, yeah. Similar to KN.
How many? What was the K? How many neighbors you took?
I think for me, I use the default K. K should be 5, I think. And I also select the I also control the correlation value. Yeah. So it's 5 like a hard threshold.
Okay. Any other questions? John, another question. So what do you think if you apply, first question is how difficult to apply your model to other data sets? For example, we can take this data set from the last year competition. Yes.
Yeah, that's a good question. I think there are no constraints or there are no obstacles if we want to transfer the method based on neural network and previous models to the other data set. I also participated in the previous competition and in the first single cell competition, and I think I do believe that their results are the same as the matrix form here. Their data are also in the matrix form, including the multi-omic part and the set sequence part. I think there will be no obstacles. Yes, we just need to change the input data, but we follow the same pre-processing and prediction steps.
Okay, but what do you think? Is it possible to make some kind of universal or semi-universal model, and then use a kind of transfer learning approach? So I mean, to train some model on big data set and only then fine-tune it for each.
Yes, it will be possible. I think it will work because I think it's like a pre-training steps, pre-training and fine-tune. I think. Yeah, I think if as long as we have like enough data set, like we have data set from human cell atlas, I think, for example, and to use human cell atlas data to train a very large model with a large set of parameters, like it should fit, should predict very well for different kind of data sets. Yeah, but the training time may be very, very long. I mean, I don't know the specific time, but it should be very long.
But you have you tried by yourself, or maybe you
no, I haven't tried
okay.
it.
And do you know maybe are there some papers on that? Haven't you looked? Are there some?
Yeah. Some papers they have tried, like they perform the pre-training for a single cell and single part. I think like it's single cell birch. I remember it's the name of their models. Yeah, I can send them to you. But they have a very strong device. They have very powerful GPUs and from one company. So I think maybe if you have such device, you can try it.
Okay, I see. And another question. So you mentioned this deep impute. Yes, instead of major, you also tried this deep impute.
Yeah, deep impute.
Yes, so it's a kind of autoencoder, as far as I understand, architecture.
Yes, it's kind of denoting autoencoder. Yeah.
So, but as far as that, in the ScanPy, in this more or less standard package, they also have kind of autoencoder, yes, for denoising, from the Fabian Pace team, DCA we call it, yes.
Yeah.
Have
I
you tried
think
it or
no, yeah. Thanks for the question. I haven't tried it. You can try it because actually we don't apply the we ignore the raw count data like this computation. But I think the organizers, they have published the raw count data set, and DCA specifically because DCA model the data based on the negative binomial distribution. Like, so we need to use the raw count as the input for DCA. Yeah, so this is one thing that we need to pay attention to. But if you have the raw count data, you can definitely apply DCA.
Okay. Thank you. Okay, thank you very much. So, do you plan to somehow continue the analysis of that data, or what are your kind of perspectives on things around that?
I think it depends like my workload. Like if I have good ideas, I can post it in the Telegram, for example. I think we can discuss it.
Okay, but yeah. So from your point of view, what can be the some possible directions which are interesting to investigate?
For me, I think I prefer to like firstly find a very kind of powerful tool for this kind of data sets. It's maybe also better than the top one participator in this competition. Yes, I think this may be a good starter here. Like to just focus on seeing this one data set and to perform a very powerful model, and then we can apply this model to other data sets and to perform the pre-training. Yeah, other approaches.
Okay, I see. Okay, thank you. Colleagues, yeah, okay, yes.
I think there's a last question like about the meeting chat part. How do you use the information about isotype control in static part, mouse and rat ILG? And I think like I'm confused about this question, but I think the organizer, they have mentioned that the rat and mouse type, we can ignore them. I think it is like a sequence control, or I think the signals, or I'm not sure, but you can search the post in the Kaggle. I think the organizers have mentioned this thing. We can ignore this information. Yeah.
Oh, yes. I see the question. Yeah. Okay, colleagues, any other questions? Please don't hesitate to ask. Or comments, suggestions, any kind of ideas.
Did you publish your feature importances analysis with SHAP values somewhere? For example, on Kaggle?
No,
really interesting.
I think I only publish it, like publish my results in Kaggle, but I don't publish it in otherwise. Yeah.
Yeah, that's really interesting. If you have time to create some public notebook, it would be really nice.
Okay, yeah. I can focus on this thing. Yeah.
Thank you, and thank you for that talk.
Okay, thanks. So, thanks. Have a nice day. Yeah.
Okay, so colleagues, thank you very much. Thank you very much, Tianyu, for making this nice presentation. So let's keep in contact, and okay, maybe for today we should finish.